The next talk is going to be by Rafal Woycicki. I hope I said that right, from Mariusz Nowacki's lab. Great, I practiced. And unless he changed his title is De-Novo sequencing of the Paramecium tetraurelia macronuclear genome using PacBio single molecule long reads for improvement of genome assembly and annotation. And I'm also, as many of you know, a fan of PacBio sequencing. So, thank you.
Thank you. Thank you for the introduction. Good evening, everyone. Thank you for the opportunity to present results of De-Novo sequencing assembly and annotation of Paramecium tetraurelia genome using PacBio technology. How I move it? Here. Okay. So the Ciliate Paramecium has two nuclei, the MAC, micronucleus, and macronucleus, which sequence was already discovered in 2006 using Sanger data, the assembly length of 72 megabase pair and consisting of about 700 scaffolds. In 2012, the strain 51, which we are using in our lab, was assembled mostly by remapping of Illumina data. The reason why we thought to De-Novo sequence again the strain 51 is that, as you can see on the histogram of the small RNA reads mapping to the genome, about 40% of is that about okay, it's not working. It's working. About 40% of 25 nucleotide small RNAs are actually not mapping to the MAC genome. On the other hand, PreQC model from SGA assembler showed us that the MAC genome can be actually about 20% larger. Expectations for the project were that we will get bigger total assembly length and a higher mapping rate of DNA and RNA reads, and that we will find new genes. So for this aim, we decided we will use third generation PacBio sequencing technology, mainly because of two facts: that with this technology we get really long reads, which can help assembling maybe still unknown repeat regions, and that the reads that we are getting from the templates which didn't go through any amplification. So we would think that we can get more uniformity of reads over the genome when there is less bias. The problem with the sequencing technology is the high error rate, even up to 20%. So it gives a necessity to make the step of error correction of raw reads before actual assembly. As you can see on the graph below, during library preparation, the double-strand DNA template to the double-strand DNA template, hairpin adapters are attached to one of those. One of those can be bound by the polymerase, and then the sequencing starts as a circle, and then a number of reads that we can get from the DNA template mostly depends on the length of this template constructed during the library preparation. Because of the error correction step, we were aiming for coverage about 100x, because we wanted to make the correction step using just PacBio sequences with the excess of those reads. Finally, we got 250x genome coverage, so thanks to the genome facility. And as you can see on the graph on the left side, the average length of the reads were 9,000 nucleotides. And as you can see on the graph on the right side, when each dot, especially when it's darker, coming to the brown color, represents more reads. Most of the reads were with the error rate about 18%. So finally, the Paramecium, the assemblies that we've made, so the starting from the correction step to the assembly. We use the Canu software, and for the correction, we used only reads longer than 4,000 nucleotides. For the assembly, the minimum overlap of 2,000 nucleotides. We are using at least 100x coverage of the raw reads, and the minimum with the minimum error rate 18%. I'm presenting here results of three assemblies and their parameters comparing to the PT51 reference. We got, starting from the most compact one, assembly 722 megabase pair and about 200 contigs, then 81 megabase pair and 900 contigs, and 96 megabase pair and 1,800 contigs. When we look at the 96 megabase pair assembly, you can see that number of the Illumina DNA reads as well as small RNA reads are largely bigger than those mapping to the reference. The problem is with the cDNA reads. Here, the drop is about 20% to 73% of the cDNA reads. It could be because of some misassemblies. So the two left assemblies, both DNA reads and cDNA reads are mapping in a little bit higher percentage, and in the 81 megabase assembly, there is also higher percentage of the small RNA reads. One thing which is worth mentioning is that with larger assembly size, as total number of mapping reads is increasing, the unique mapping reads are decreasing, which could be indicator of the fact that in Paramecium genome the paralogous sequences content can be even larger. So when we align new assembly contigs to the reference scaffolds, we can see that the 72, the most compact assembly, is mostly contained all in the reference assembly, in the reference assembly, but it's missing some sequences. Then in the 81 megabase per assembly, there are also smaller part of the sequences missing, but this assembly contains also sequences absent in the reference assembly. On this plot, here is the plot of the contig length versus GC content of the reference and two assemblies, and distribution is similar. While you can see that new assembly contains longer contigs, and there is one contig which is about 500,000 nucleotides longer than the longest contig in the reference. When we go deeper analyze this contig, we see that there is misassembly. So when we align the longest contig from the 81 megabase assembly to the longest contig from the reference, the similarity ends at 753,000 nucleotides. When we map the back corrected reads to this contig and Illumina reads to this contig, we see that at the position of end of the similarity there is drop of both Illumina and PacBio corrected reads. When we zoom in this region, drop in the PacBio reads goes down to 3 reads. And Illumina reads, with Illumina reads, there is no those are paired ends reads, but there is no coverage neither with reads itself, or the region is not spanned other that's in the other regions shown here. So because of the high error rate of the PacBio reads after assembly, there is important step of looking for errors, finding errors, and polishing the assembly to correct for those errors. There are two ways. So we can use PacBio raw reads or Illumina reads. We used 200x coverage of Illumina paired-end reads. And from the statistics table, from the results table, we can see that most of the errors are single nucleotide indels, and then the error rate in the PacBio assembly is of about 1 error per 10,000 nucleotides. So the next step was to check if in the new assembly we can find any new genes. So De-Novo gene prediction was made with Augustus using training, of course, and hints generated from 100 million Illumina cDNA reads. And for 81 megabase genome, we found about 39,800 genes. Proteins, those proteins were blasted with blast P at the eValue threshold 1e minus 0.5 to the database of reference proteins. And I'm sorry, this is here on the graph at the top. That's the length distribution of proteins, new predicted proteins and reference proteins. It looks similar. And after blasting those two databases, about 2,500 proteins didn't find any hit. But when we plot protein length of those which found the hit to those which didn't find the hit, the blue color, we see that drop in number of those proteins which didn't find the hit is about 200 nucleotides. So we chosen the threshold of nucleotides, amino acids. So we chosen the threshold of 200 amino acids as the one to say, okay, that could be maybe about 300 new genes not found in the reference. So this work is still in progress, but quickly to check if those predicted proteins can code, can be coding. We aligned those to the UniProt database of proteins and to the FAM database of domain, and the result was that it found from 290 to 94 proteins with hits in UniProt and 21 proteins in hits with FAM database. So in summary, according to the results that I've shown, the Paramecium genome can still be larger than this 82 megabase and can consist of more paralogous regions. That possibly, thanks to long PacBio reads, most of the coding part of the genome was assembled, and PacBio self-correctness algorithms are sufficient to correct high row PacBio error read errors. And there is still need to correct for misassemblies, for example, by comparing Illumina and PacBio row reads coverage in context. And thanks to the De-Novo PacBio draft assemblies, as well as more than 100 million cDNA Illumina reads, it was possible to improve gene prediction and possibly discover at least 100 new coding genes in Paramecium genome. This work is in progress, and also the assembly is still in progress. I would like to thank current and former lab members, and also I cannot show. So on the left, the people who were involved in the project. So Christina, Aditi, Kerstin, Estienne, Mariusz, and Simran, and the rest people from the lab. Thank you for your attention.
Please come use the microphones for questions. Actually, I'll start with the questions. Since you're in a great position of having so much PacBio data that is so much high coverage from the same genes, are you also looking at DNA modifications?
No.
Also sort of related to that, so you said you'd use, I think, 250x coverage for the PacBio, which seems very high. So I'm just wondering, like, why you went with that amount. Like, is it?
No, yeah. So that's we were aiming for 100x. And that's what we wanted from the Genome facility.
Oh,
And then they give us 250.
That's a good
Just
deal.
like that.
Yeah.
Yeah.
So 250x, I guess, is the average coverage for the whole genome that you got?
Yes, of course.
So if you look only at the new part of the genome, let's say the sequences that you have added to the pre-existing assembly, what is the coverage that you get there?
I cannot say like this because 250x coverage is of all the reads from, let's say 75x coverage. So what I was taking for the assembly was reads from 82. From having less than 18% error or less than 15% error. So I was taking about 100x coverage or 180x coverage. I was not taking all the reads because I wanted to take mostly the reads which have less error rate, and then I have still the coverage. But but answering.
No, but in fact, because you showed a figure where you actually connect two scaffolds by a region where you have very few reads.
Yeah.
Just like this.
So I'm checking this in more detail now. But when I was checking at the few contexts, there have been maybe few instances of coverage when actually the number of reads were dropping below 10. Number of reads below 10. It's few instances. Mostly it was more than 30x in the of coverage of context.
So I was wondering if you think that the 236, if I remember correctly, contigs that you have, how many of them are full-size chromosome? I was surprised to see like very few of them are telomeric repeats at the end in your table.
This could be also, so I cannot say this neither now. Yeah, that's low, but this can be also because of some errors. I mean, this value, I think it's from results before I was polishing.
Okay.
I have, but
because I think
it's low right here.
That's roughly the number of chromosomes that we expect. I think
Yeah.
so. Can I ask one more?
We have extra time because the talk afterwards.
Okay, so I have two very practical questions because we might be interested in doing PacBio someday. First, did you try to do subsampling to see what coverage would be just necessary? I assume that 250 is an overkill, but where do you reach the plateau? Will you have had roughly the same results with like 50, 80, 100x, and then what the corresponding cost would be?
No, I was not doing some sampling because the first step before I can really use those reads was error correction, and this error correction step I think took like half of the time that I was working on this because there were problems with algorithms. So, no.
Can you? For
the PacBio, because I mean, in the PacBio first step is error correction. You need excess of reads to error correct, and then this 100x coverage that I'm taking of raw reads, half of this coverage is used for error correction. Then I'm left with maybe half or even less than half.
Okay, thanks.
So it's hard to answer this. It's you cannot say that you can use this read and subsample. Yeah.
So of the predicted 300 new genes that you've identified through the sequencing project, have you looked to see whether they're what I would define as a novel gene or they're the product of the gene duplication, so they're paralogs of genes that have been previously identified?
Okay, so I have to say that only what... okay,
I got you.
Yeah.
Okay.
Answer the question, no, I didn't have time for this. The only thing which I can say that the hits from the UniProt There is no hit. There are hits for tetrahymena. There are hits for Stauronekia. There are hits for Auxitricha, and there are also hits for plants and animals. But yeah, I mean, none for Paramecium, of course.
Okay.
But yeah.
Okay, thank you.
All right, I'll thank Rafal again.
Thank you.